Scientists have many tools available for them to ensure that they conduct accurate and the best possible research that they can. For biologists, perhaps one of the most important tools at their disposal is a microscope. A microscope is simply a tool that is used to make small things look bigger and be able to see them well. The first compound microscope was invented in 1595. Any type of lens that makes things look larger is technically considered a microscope. A compound microscope is one that contains two or more lenses. Now, this first microscope was invented by a father and son named Hans and Zacharias Jensen, and their microscope looked very little like what we have today. It was basically a straight tube that had a lens at opposite ends. You looked in one end, and you could put the object at the other, and you could see with a little bit of magnification. The images that you were able to see through this microscope were not particularly good. They were fairly blurry, and they had very little magnification. Looking at the microscopes that we have today, we have a modern compound light microscope. It is important that we add in the term light because light is being used to create the image opposed to something else. There are other types of microscopes that create images in other ways. Compared to the first microscope, the modern compound light microscope has some fairly good clear views up to 1,500 times magnification. That's significantly more magnification than the very first compound microscope. It does, however, have its limitations. One of the limitations is that it can only magnify things up to 1,500 times. There are other types of microscopes that can literally magnify things hundreds of thousands of times their actual size. Another limitation is that whatever you're looking at needs to be thin enough for light to shine through the object. And so that's how the image is created. These are some examples of specimens taken with a compound light microscope. One of the benefits of using a compound light microscope, you can observe live specimens and you can see things in color the way that they actually are. Images from some other microscopes, you get some really neat images, but they tend to lack color because you need to have light to see color, but you've got significantly more magnification with these images. Some types of microscope images do have color, but the color is added in simply for contrast, and it's not the actual color that the objects are. With our compound light microscope, there's a number of very important parts to it that are essential for its function. Starting at the very top, you have the ocular lens. This is where you look into the microscope, and it provides 10 times magnification on our microscopes. You have a number of objective lenses, the smallest of which is the low power objective lens. It by itself provides 4 times magnification. Compound light microscopes usually have at least 2 objective lenses. The high power objective lens is found over the object as well, and it provides 40 times magnification. All these lenses are supported and held up over the object that you're looking at by the arm of the microscope. The specimen that you're observing gets placed on the stage and is held in place by stage clips. Underneath the stage is a diaphragm. There are 2 types of diaphragms. One is called a disc diaphragm, a circular piece of plastic with different sized holes cut in it. The other is called an iris diaphragm, which has a lever on it. Regardless of what type of diaphragm it is, it's intended to control the amount of light that is shining through the specimen, so you can make the light brighter or dimmer depending on your needs. You of course have to have a light source. On most modern compound light microscopes, this is provided by some kind of lamp. There are some that exist still that have a mirror that redirects light from outside the microscope through the specimen. The coarse focus moves the stage up and down very rapidly in order to adjust the clarity of your view of the image as you look into the ocular lens. The fine focus, which is the smaller of the 2 knobs, it moves the stage up and down very slowly, and this is to very carefully and precisely adjust the clarity of the image that you're looking at. The very bottom of the microscope is called the base, and it basically holds everything together and supports the whole microscope. The base, in combination with the arm of the microscope, are the two places that you hold on to it when you're carrying it from one place to another. Now, I mentioned the magnification of the individual lenses. The ocular lens has a 10 times magnification, the low power objective lens has a 4 times magnification, and the high power objective lens has a 40 times objective magnification. But you're working with these two lenses at the same time, and so you need to be able to calculate the total magnification of the image that you're seeing. Remember that calculating total magnification is done by taking the ocular lens magnification and multiplying it times the magnification of the objective lens that you're currently using. When you look into the microscope, the area that you see is known as the field of view. On low magnification, you can see a fairly wide area. This is why you always start on low power, so that you can find the object that you're trying to look at. As you go to higher and higher magnification, it's kind of like you get closer and closer to the object. You zoom in, and so your field of view becomes smaller. You see less of the area around the object, and you're able to see in more detail the particular object that you're looking at. When using a microscope, you're going to start on low power with the stage all the way up. You need to have your specimen and put it on the stage. Make sure that it's secured under the stage clips. You're going to look through the ocular lens and adjust the coarse focus until you're able to see the specimen that you're looking at. The thing you have to remember is that on low power, the object you're looking at may still be fairly small. Once you've found the object, you may want to make sure that it's centered in your field of view. Remember that when you go to the next higher magnification, you're going to be zooming in, and if it's not centered, when you zoom in to the next higher magnification, it may be outside your field of view. When you switch to the next higher magnification, you're going to adjust the fine focus. Remember that you should never touch the coarse focus unless you're on low power magnification. Once you've adjusted the fine focus, if you need to go to a higher magnification, you can repeat steps 4 and 5, centering the object and then switching to the next higher power and using the fine focus. So those are the basics of the microscopes that we make use of. Be familiar with them. We will be using microscopes throughout the year for a variety of different activities and lab work.